In this video, we're going to dig deeper into the process of regenerating or producing ATP from ADP and an inorganic phosphate group. As a reminder, this process is an anabolic endergonic reaction, so energy is required in order to build adenosine triphosphate from adenosine diphosphate and an inorganic phosphate group. Okay, so where does this energy come from? We've already talked about how ultimately the energy to power this endergonic reaction comes from the exergonic catabolism, meaning breakdown, of glucose and other food molecules in the process of cellular respiration. But now we're going to dive into the process that directly accomplishes the production of ATP, which is chemiosmosis. In a way, we are kind of jumping to the punchline of cellular respiration. And we're doing that because it's the most important part. It's the payoff in which we get the big bonus of producing many ATP molecules. All right, let's take a closer look at the enzyme that accomplishes the reaction of synthesizing adenosine triphosphate from ADP and inorganic phosphate. The enzyme, which is a large quaternary protein complex, is called ATP synthase. Remember how enzymes are cool because their name tells you what they do? Well, here's a perfect example. ATP synthase synthesizes ATP. ATP synthase consists of a number of subunits that are embedded in a membrane, forming a pore through which hydrogen ions, otherwise known as protons, can flow. This powers the part of ATP synthase that forms a bond between ADP and an inorganic phosphate group. That's shown here in red and pink. This part actually turns as protons flow down their concentration gradient through the pore part of ATP synthase, which is on the top, powering the endergonic reaction that regenerates ATP. We sometimes refer to ATP synthase as a molecular motor because the whole complex is actually turning as this reaction takes place. We can find ATP synthase embedded in various membranes, the inner membrane of the mitochondrion most famously, but also the thylakoid membranes of chloroplasts and the plasma membrane of prokaryotes such as bacteria. The process of building ATP is again endergonic, so we need a supply of energy. In other words, we need to couple this process to an exergonic reaction that releases energy. And that, in this case, is going to be the diffusion of hydrogen ions flowing down their gradient. Recall that diffusion is the flow of substances down their concentration gradient, in other words, from an area of high concentration to an area of low concentration. And diffusion is a process that does not require energy. In fact, it releases energy. Chemiosmosis generates more than 80% of the ATP made during the breakdown of glucose and other food molecules. It also functions in the chloroplast to produce ATP as part of the light-dependent reactions of photosynthesis, as well as in bacterial cells, which is not shown on this slide. So here we're comparing the process in the mitochondria and the chloroplast. And what you'll notice is that the process is the same. We have an area with a high concentration of hydrogen ions or protons, and we have an area with a low concentration of hydrogen ions or protons. These hydrogen ions flow through the pore part of ATP synthase, turning the molecular motor part, which directly catalyzes the addition of the inorganic phosphate group onto adenosine diphosphate, which produces adenosine triphosphate. Okay, so for chemiosmosis to take place, we are, of course, going to maintain the gradient or concentration difference of hydrogen ions on either side of whatever membrane ATP synthase is embedded in. And we're going to cover how that happens in future videos.